Okay, guys, let us start our today class. Okay, it's neuromuscular junctions. So what are the learning outcomes that we need to learn from these subtopics? Okay, first of all, we need to describe the role of the neuromuscular junction, transverse system tubules, or in our syllabus, we term it as T tubules. Oops, sorry. Okay, transverse system tubules or T tubules, and then a special reticulum, okay, or endoplasmic reticulum, or smooth ER is termed as sarcoplasmic reticulum in the stimulating contraction of the striated muscle. So we're going to differentiate between three kinds of muscle cells. One is striated muscle, or is termed as the skeletal muscle, the muscle that attached to your skeleton. This one is called a skeletal muscle. And we do have the smooth muscle as well as the cardiac muscle. So we're going to look at what are the differences between these three kinds of muscle cells. And then we need to describe the ultrastructure of the striated muscle, reference to the functional unit of the muscle cells called sarcomere, the sarcomere. Okay, the structure of sarcomere, where we're going to look at quite a number of alphabets, for example, the A, the I, the H, Z, and M. Okay, so we need to know what they are referring to. And the last one, we're going to look at what we call the sliding filaments model, or the muscular contractions, involve the role of these few proteins. Okay, one is a troponin, tropomyosin, okay, calcium ions, and also the ATP. And beside these two, we're going to look at few kinds of the myofiber, okay, termed as actin filaments, as well as the myosin. Yeah, so students found it quite confused because too many things. You have the troponin, tropomyosin, actin filament, actin molecule, and myosin head. So you have to be careful when you learn these topics. Okay, so we started with the types of muscle cell first. Okay, so muscle cells, also known as the myocytes, the proteins, okay, the special oxygen storage proteins inside the muscle cells, also known as myoglobins. Okay, it's oxygen storage. So myocyte is the cells, okay, muscle cells that make up the muscle tissue, and we do know that we have three kinds of the muscle cells. We have striated muscle cells, cardiac muscle cells, and smooth muscle cells. Okay, so out of these three, cardiac muscle and smooth muscle, they are involuntary. Now, what's mean involuntary? Involuntary basically means that we do not need to control them, okay, by using our conscious mind. For example, cardiac muscle, okay, you do not need to actually tell them, oh, contract, oh, now relax, now contract, now relax, right? If you need to tell them what to do, what will happen? One day you forget, or we forget to tell them to contract, and then forever we don't need to tell them to contract anymore, okay, or pump, right? So cardiac muscle, smooth muscle as well. Smooth muscle, particularly when you breathe in and breathe out the air, they control the diameter. So smooth muscle generally control the diameter of what? Control the diameter of arterial. We learn in thermoregulations, right, for vasodilation, vessel constructions. Okay, vessel constrictions, okay, and then also in our windpipes, right, our bronchus, and also the trachea, we do have the smooth muscle to regulate the diameter, and also smooth muscle involved in the gastrointestinal tract when we are going to look at this. We don't learn this, but the peristalsis between the two kinds of muscle cells, the smooth muscle cells, and those muscle cells we do not need to regulate or call or tell them what to do. Okay, so but our focus for today's lessons will be on striated muscle, where we need to use our conscious regions, okay, to control their movement. For example, you want to do, okay, for example, you want to jump, you want to talk, you want to flex your muscle, you want to remove your arm. So those require your conscious regions of your brain to instruct. Okay, so basically the action potential will be sent from your brain through the output through your muscle cells, through the motor neurons. Okay, so we look at what are the differences between this striated muscle, cardiac muscle, and smooth muscle. So when talk about striated muscle, they are voluntary. Voluntary means that we need our brain to control them. So generally used for locomotions. Locomotions means that the movement. Okay, cardiac muscle, as I said, that is involuntary, means that we do not need to use our conscious region of the mind to control them. Okay, found only inside the heart. And cardiac muscle also special. They are myogenic as well. Now, what is myogenic? Myogenic basically means that they're involuntary. They can actually contract by their own, and they do not need any nerve impulses, even though nerve impulses or what we call this hormones, and like for example our adrenalines, okay, hormones, our action potential may have an effect on the cardiac muscle, but on its own, the cardiac muscle, if I take out your cardiac muscle, they can actually, without any stimulus, without any impulses, action potential, they still can contract. So this we call myogenics. Okay, they do not need to have any impulses. Okay, in this case, here. And the last one we talk about smooth muscle. So smooth muscle, again, involuntary, always found in the wall of the organs, such as the trachea, bronchi, esophagus. Okay, blood vessel, for example, arterial, so that it can help us to regulate. Most of them actually help us to regulate the diameter of the tubule, okay, most of the time. So you look at the structure. So look at the structures, skeletal muscle. Skeletal muscle also known as striated muscle because of the striations, okay, you can see that. If I enlarge it, can you see that? Oh, sorry, the line. So this line is known as the striation. Okay, so they are more to cylindrical shape, okay? The shape is a cylinder. So how about smooth muscle? If you look at the smooth muscle is more to spindle shape. Okay, smooth muscle. And cardiac muscle, you look at cardiac muscle, they are interconnected. Something similar to skeletal muscle, they have the striation as well, but they are interconnected. Okay, so they have the intercalating disc. And that's very, very important why they need to have the connections like this, connected, means that because we need to know one thing because of this cardiac muscle, when they are myogenics, one patch of them contract, like for example, the SAN, sinoatrium nodes contract. So you're going to trigger the rest of the cardiac muscle to contract, okay, by sending out this what we call the electrical excitation wave. So electrical excitation wave is not the action potential. Are you clear? It's more to something like electric. So it means that when the contraction of one patch of the cells, you're going to trigger the rest of the cell to contract. So totally different from the skeletal muscle. Okay. So just for information only, our focus will be here, striation muscle, striated muscle only. So appearance, okay, appearance under the light microscope, you can see that there are stripes, means that there are striations at a regular interval, same as the cardiac muscle. Smooth muscle, they don't have the striation. In terms of the cell structure, striated muscle has a multi-nucleated, or we call it syncytium. Syncytium basically means that more than one nucleus. So it means that they are multi-nucleated cells. Okay. So cardiac muscle, they are uninucleated, means only one nucleus per cell, but they join by the intercalated disc. This is very important to make sure that one cell contract is going to cause the rest of the cell to contract at the same time. Smooth muscle, uninucleated. Uninucleated basically means that one cell has one nucleus only. In terms of the shape of the cell, striated muscle, cylinder. Okay, cardiac muscle also a bit a cylinder, but shorter. And they have the branches that connect to adjacent cells, but smooth muscle appear as a tapering end. Okay. So organization of contractile proteins. So it's a parallel bundle of myofiber. This is very important. We're going to learn this one, okay, in detail later. And how about cardiac muscle? Also organized as a parallel bundle of the myofiber, but smooth muscle, the contractile protein are not organized into the myofiber. Okay. How about distributions? So always remember, striated muscle, also known as a skeletal muscle. So it means that the muscle cells that attach to the skeleton. Cardiac muscle at heart, and smooth muscle generally at the tubular structure. As I said, that the blood vessel, the airway, okay, those tube, then you can see that we have a lot of this smooth muscle. So in terms of control, neurogenics means that they need the action potential. Right down. Myogenic means no need action potential. Yeah, so there's a difference between the myogenics and also neurogenics. Neuro means neuron, so needs to have the neuromuscular junction there. Okay, So now we're zooming into the structure of the striated muscle now. So the functional unit, as I said that just now, okay, we have seen this, is a muscle tissue that features repeating functional unit known as a sarcomere. So it means that you can see that cylindrical shape. Okay, so at regular interval, you will see something like this. Something like this. So this is one sarcomere, okay, one sarcomere in this myofiber, okay. So each muscle fiber is surrounded by the plasma membrane, okay. Their cells, they should have the plasma membrane, but this plasma membrane, we give them a special name called sarcolemma. Sarco means muscle, okay. Sarco means muscle, lemma here means membrane. So that's why it's a membranes of the muscle cells. So cytoplasm, we also give them a special name, okay? They are cytoplasm. We also call them as a sarcoplasm, okay? Is the cytoplasm of the fiber, okay, or the muscle cells. So there are high number of mitochondria highlights. This is very, very important because for muscle contractions, you need to have ATP, okay? And abundance of oxygen binding protein known as myoglobin that we learn in the AS topics. Okay, myoglobin act as the O2 storage. So it means that at rest, when you breathe in the air that release, the oxygen that released by the hemoglobin, not all will be used for respiration. So some of them will be will bind to the myoglobins and store as the oxymyoglobin. So oxymyoglobin is special because they only release, okay, the oxygens when the hemoglobin cannot meet the demands, particularly when we carry out the vigorous activities, okay? So the sarcolemma has a many deep infolding into the interior of the fiber known as a T-tubule, highlights, okay? We learn it as T-tubules or transverse tubule or transverse system tubule. And this run close to the sarcoplasmic reticulum. So it's a special endoplasmic reticulum, or more precisely, it's a special type of SER known as sarcoplasmic reticulum. So what's the function of sarcoplasmic reticulum? So generally, sarcoplasmic reticulum act as the calcium storage site for calcium ions. Okay, so let us look at the diagram before we continue, because a lot of the term now. I'll enlarge this diagram. Okay, so you can see that this is one muscle cell. This is one muscle cell. So this is one sarcomere. Okay, one sarcomere. Okay, sarcomere. So one sarcomere, and okay. So what we're going to do here is, if you look at this, they as I say that they are multi-nucleated. You can see that one cells, they have many nuclei. So that's why we call it multi-nucleated. So the cytoplasm of the cells is known, a muscle cell is known as sarcoplasm, and the membrane, can you see that the membrane is known as sarcolemma? Okay, so inside here you have a lot of protein fiber involved in the muscle contractions. We term them as a myofiber. So enlarge this part, you can see that this is one myofiber. Can you see that? One myofiber. So they are cylindrical, so we can pack them. Can you see that? We can pack them. So this myofiber is wrapped by a special kind of the smooth endoplasmic reticulum known as sarcoplasmic reticulum. You try to imagine, okay, you try to imagine this myofiber wrapped by who? Wrapped by this sarcoplasmic reticulum. Okay, so what's so special about this sarcoplasmic reticulum? Sarcoplasmic reticulum actually serves one very important function. They are calcium ion storage. They store calcium ion. Are you clear? Which is very important later for the muscle contractions. Okay, that's what. Second, if you look at this sarcoplasmic reticulum, you have something like a rubber band that tied them, right? It's something like a rubber band, right? Tied them together. Can you see that? So this yellowish color here, like the rubber band that tied them around this myofiber, is known as the T-tubule. Okay, just now we learned about this. Sarcolemma is not a smooth surface. They are perforated by the pore, and this pore continue into this T-tubule. Can you see that? This pore continue into these T-tubules. Okay, I'm going to show you one diagram now, not here. Wait, guys. Now, can you see that, guys? So this is the muscle myofiber, this partner, myofiber. The one I highlight now, myofiber. So myofiber actually enclosed or wrapped by this, the bit blue color here, the thing. So this blue color thing is known as the Sarcoplasmic reticulum, which act as the calcium ion storage. And then we do have this, what we call the plasma membrane. Can you see that plasma membrane? So this plasma membrane is known as a sarcolemma. Okay, the plasma membrane is known as sarcolemma. And this sarcolemma is not smooth like this, but perforated by the pore. And this pore continue, okay, into this, what we call the T tubule. Now, what's the function of T tubule? Very important. In order for myofiber or the sarcomere to become shorter, to contract the muscle, we need to have action potential. But if without the T tubule, what will happen? Your action potential will transmit along the sarcolemma. It won't enter into the cell. Correct not? Your action potential generate, stimulate another region, stimulate. So depolarize, depolarize, depolarize. If you continue like this, the action potential. So it won't cause any effect to the muscle fiber or myofiber. So what is the function of T tubule? T tubule actually conduct the action potential from the sarcolemma. Can you see that? All the way enter into the T tubules, and then it's going to cause this calcium ion to be released, and calcium ion will cause the shortening of the sarcomere. Can you see that? So this is very important again to see that T tubule is a very, very important structure, allow the transmission of the action potential from the sarcolemma all the way into the psychoplasm, where they're going to trigger the release of the calcium ion later. We're going to learn this in detail. Okay, so why it is perforated? So questions sometimes ask, what are the fluids that fill in this T tubule? Because they're exposed to the tissue fluid, right? So it means that the fluid that inside the T tubule will be similar to the tissue fluids. So it means that outside will be high with the sodium ion. Can you see that? High with the sodium ion. So inside the cells will be low with the sodium ion. So any depolarization will cause the sodium ions to enter into the psychoplasm. Okay, yeah? So come back to this diagram now. So get the idea now, get the idea. So basically, we do have these few terms that you need to use generally. So first, the cytoplasm of the muscle cell is known as sarcoplasm. The membranes of the muscle cell is known as sarcolemma. This sarcolemma is not continuous because they have this pore or perforated and connected to the T tubule. And this T tubule actually will go down and then associate with the sarcoplasmic reticulum, which act as the calcium ion storage. And this sarcoplasmic reticulum actually associate with the myofiber. So now we're going to zoom in into this myofiber by looking into these two kinds of the muscle fiber, two kinds of proteins. One is called thin filaments, or later I will call it as a thin actin filament. And thick filament, also known as a thick myosin filament. Okay, so they are fibrous proteins, guys. They are fibrous proteins, okay, in general. Okay, so let us zoom into this myofiber now. So myofiber, they compose a long fibrous protein known as the myosin and actin. Myosin and actin. So this protein are organized into the thick and thin filaments known as a biofilaments. Okay, and they arrange in a section known as sarcomere. So let's look at the thick filaments first. Okay, thick filaments. So these thick filaments consist of several hundred molecules of myosin shaped like the golf clubs. Okay, so it's the shape something like this. Okay, so this part is called as a rod. This part is called as a head. So myosin rods and myosin heads. And because of this, later they will form a thick myosin filament when several hundreds of molecules actually join together. And each myosin molecule consists of two heavy myosin chains wound around together to form a rod portions. lying parallel to the myosin, myofilaments, and two myosin heads. So this is one, okay, parallel. So we can have another one. So it will be something like this, okay, but they're intertwined together, okay? So myosin head attached to the rod, so head attached to the rod portions by a hinged region. So give the flexibility. Always remember, like antibody, the hinged region, so give the flexibility. Okay, and the heads highlight, they have this ATPase activity. So basically means that they have ability to hydrolyze the ATP to become ADP plus PI. Okay. So what they do, their functions, basically during the muscle contractions, these myosin heads will bind to this myosin head binding site. Okay, myosin head binding site on the active molecule to form the cross bridge. So don't worry about this part. Later, when you look at the mechanism, you will be able to view it. Okay, just now you can see that this myosin head can bind to the myosin head binding site, okay, at the actin molecule. So look at this structure, guys. So you can see that you have two heavy chains intertwined together to form this helical structure, double helix structure with the hinged regions. Okay, the hinged regions. So this one something like golf clubs, right? Something like golf clubs. So this is a head and this is the rod. Okay, so the heads have this ATP binding site. So ATP can bind. When ATP binds, then it's going to cause a series of reactions. Okay, and the heads will have this what we call the ATPase activity. Okay, so we can see that because of this structure, it forms a thick filaments, thick filaments or myosin thick filaments. Okay, how about thin filaments? Thin filaments composed of three major protein highlights: F-actin, F stands for fibrous, tropomyosin, and troponin. Okay, so we have these three highlights you need to know. So what's so important about F-actin? So the major components of the thin filament is this fibrous actins. So this fibrous actin also form a double helix, extending the length of the myofilaments, okay, attached at either ends of the sarcomere. So each strand of F-actin, okay, autocorrect, sorry. F-actins, okay, is composed by actin monomer, each of which have the myosin binding sites, and it provides this site for the binding of the myosin heads during muscle contractions. Okay, so let us look at this protein. Now, what is the G-actin? Just now we have F-actin. F-actin refer to the fibrous, so it means that this is the fibrous. Can you see that? The fibrous. Okay, two of them intertwine together to form this double helix again. Are you clear? Double helix. Okay, and each of them actually formed by the G-actin. G stands for globular. Okay, globular. So it means that it's not one protein. It's not one protein all the way. If you compare to the myosin, myosins is a whole continuous protein. But this one actually, they have the subunit. So they have the globular actin joined together, 1, 2, 3, 4, 5, 6, joined together to form the fibrous actin. And each of the actin molecule, can you see these are yellow color? The yellow color right now is the attachment site, or we call it as the myosin binding site. Or for me, I prefer to call it as a myosin head binding site, where later the myosin head can bind to this binding site and form the cross bridge. Okay. Now, during the muscle relaxation, we don't want to have muscle contraction. So it's something like you don't want to have the cross bridge. If you don't want to have a cross bridge, then we cover it. Correct or not? If you don't want anything to bind to this binding site, we cover it. So how we cover it? We cover it by using the tropomyosin. Can you see that? Something like a rope. So this protein is a fibrous protein that covers these binding sites. Can you see that? So tropomyosin. And this tropomyosin linked with one protein known as troponin. And the special about this troponin actually has the calcium ion binding site. Okay, they have this calcium ion binding site. So what will happen here is when the calcium ion bind to troponin, just now we briefly I've gone through with you guys saying that when you have the actual potential enter into the T tubule, it will cause the calcium ion to be released from the sarcoplasmic reticulum, right? So these calcium ions will bind to the troponin. When they bind to the troponin, it will cause the troponin to change its precise three-dimensional shape. And because of troponin bind to this tropomyosin, it will cause the displacements of the tropomyosin and hence expose the myosin head binding site. So this is the entire thing here is very thin. So that's why we call them as the thin filaments or thin actin filaments. Are you guys clear about this now? Okay. So we have thin filaments. We have the thick filaments. So thick filaments, they have this rod structure and the head structure. And the head structure, okay, or have this ATPase activity. So ATP can bind to the head of the myosins, okay, or myosin heads. And for the thin actin filaments consists of 3 kinds of protein. So the fibrous actin or F-actin, and then F-actin consists of the monomer known as the G-actin. G stands for globular. So this G-actin actually join together to form the F-actin. And each molecule of G-actin consists of or have this or we call the myosin head binding site. And when muscle relaxation take place, it is or they are covered by by this or we call the tropomyosin. And tropomyosin join or actually associate with the troponin. So this troponin has the calcium ion binding site. So it means when the calcium ion bind to the troponin, troponin change the shape. And because troponin bind to the tropomyosin, when the troponin change the shape, it will cause the tropomyosin to displace, or move. So when the movement of the tropomyosin is going to expose, I'm going to expose this myosin head binding site, allow the myosin head to bind, and then you can see that the muscle contraction actually take place. So how we arrange these actins and myosin filaments? So they are arranged in a structure known as the sarcomere. Okay, they are arranged in a sarcomere. So if you look at it under the light microscope, the myofiber has a light and dark band known as the striations. So if examining the sarcomere, then they are when they are relaxed, show the sarcomere extend between 2 dark lines known as the Z line. So Z means between. Okay, so if you look at this, I'm going to draw it out now. So this is the sarcomere. So entire thing is known as the sarcomere. Sorry. Okay, so the whole length. So sarcomere is characterized by the distance between 2 Z line. Okay, can you see that? Between 2 Z line. Okay. So what is the function of Z line? Z line actually is something like a protein, it's a holder. Always remember, it's a line, you may view it. So means that if you are having the structure like 3-dimensional structure, you can see that this is a Z line. This is a Z line. It's something like a plate. So sometimes they also call it as a disc. Can you see that? Z line. So the distance between those two, is known as the sarcomere. So this Z line is very important because Z lines are going to hold the thin actin filaments. So means that you can see that the thin actin filament, too long. Okay, thin actin filament, thin actin filament, thin actin filament. Okay, so I use this. So this is a thin actin filament. Okay, use different color. Okay, so at the middle of the Z line, at the middle of the Z lines, you're going to have another line, known as middle, right? At the middle. So known as the M line. Okay, we have Z line and M line. What's the function of M line? M line actually going to hold the thick myosin filament. So you can see that thick myosin filament, thick myosin filaments, thick myosin filament. Are you clear? Can I see that? Okay, so look at the structure. We have the Z line. So the distance between the Z line actually is Z line. This is the length of the one sarcomere. Are you clear? Okay. So then you can see that at the center, middle, right? Middle. So middle M. Okay. So middle actually is the one that hold the myosin filaments, thick myosin filaments. So I label here. Sorry. Thick myosin filament. Okay. Can I see that? Okay. So now, this is one sarcomere. This is one sarcomere. But if I have many sarcomere joined together, then you're going to see as the zone. You will see the zone. Okay. So let me draw it out now. You can see that. So you have one Z line here. Okay, I draw 3 sarcomere. Okay, 3 sarcomere. So we know that this is Z line, Z line, Z line, Z line. Okay, so this is one sarcomere, right? Okay, one sarcomere. So Z line actually have the thin filament. Okay, Z line hold thin filament. Okay, then we do have the M line. In between, right? M line, M line, M line. So this M line, M line, M line. Okay, M line hold what? M line hold the thick filaments. Okay, thick filaments, thick filaments, thick filaments, thick filament, thick filament. So can you see that it forms a zone? It forms a zone. So if I color it, you can see that you have the zone that's lighter in color. This part. Can you see that? Lighter in color. Lighter in the color. Okay, so this lightest in color zone here is known as the I zone. Okay, it's known as the I zone. Okay, so I zone, if you look at I zone only has the thin actin filament. Okay, only has a thin actin filament. Are you clear? And second, you can see that I have the zone with a darker color here. Can you see that? Darker color zone. So this darker color zone actually is the length of the myosin. So this dark color zone is known as the A zone or A band. I band or I zone, A band or A zone. Okay. Can you see that? A band. So A band consists of what? A band actually is the entire length of the myosin head. It's a myosin filaments, okay? Entire myosin filaments. But in between of the A band, you have this slightly lighter color zone here, right? Can you see that? Are you clear? So this zone is known as the H zone or H band. Okay, either H zones or H band. Can you see that? Okay. So we have I band, A band, and also the H zone. Okay. Okay, I put it band better. Okay, so H zone only has the thick filament, thick myosin filament. Okay, so let us look at this diagram now. So can you see that the zone with the thin filaments only, ignore the connecting, connectings. Okay, so you can see that this zone we have thin filaments only, so it's an I-band. Okay, it's the I-band. So it means the thin filaments will anchor. Can you see the anchor on what? Anchor on the Z-dis of Z line. Okay, and then after that you can see that the length, this entire line is called A-band. Can you see that? Entire line is called A-band. So A-bands will have the myosin filaments and also the thin filaments. Can you see that? It's a mixture. Okay, at two end there, you have the mixture of the thick filaments and also the thin actin filament. Can you see that? Okay, so the entire structure called A-band. So within the A-band, you have a lighter zone that there is no actin filaments. Can you see that? No actin filaments, known as H zone. So right now, I-band only has, okay, I-band only has thin filament. H zone only have thick filament only. For A-band, then at two sides, you have the mixture of both. Are you clear? For A-band, you have the mixture. I mean, at both sides. Center side, no. Center side only, right? Of the thick filaments. Okay. So now try to figure out what is A. Try to figure out what's A. If you look at A, this is one. Okay, Z line. This is another Z line. So B actually is a Z line. Okay, B is a Z line. So A is the entire length of the sarcomere. A is entire length of the sarcomere. Okay, B is the Z line. Can? Now how about C? C is the line, the center line here, right? For the thick filament. Center line for the thick filament. So the C actually is the, you look at the C actually refer to the M line. Okay? C is the M line. So D, we know that these are thin filaments, right? The lighter zone. So lightest zone actually is the I bands. Okay, I band. Darker band is known as the A band. Okay, then the small, tiny, a little bit lighter in color. So this one is known as the H zone. So during muscle contractions, okay, so the sarcomere become shorter. Okay, during muscle contractions, the entire sarcomere becomes shorter. But you look at this, okay, if you look at this, okay, structure, the A band. So the A band is the length of the myosin head, myosin. So means that the length won't change. So what we change? We change the distance or the length of the I band, and also the H band becomes shorter. When during muscle contraction, okay, H zone and the I band become shorter. A band actually remain the same. Okay, A band remain the same. So now entire sarcomere become shorter. Okay. So what is A? A is named as the sarcomere. So sarcomere extend between the two dark lines known as the Z line. What happened during muscle contractions? The entire length of sarcomere now become shorter, right? Because Z line move closer. So how about the Z line? Z line actually is this act as an anchoring point for the thin actin filaments. So there's no change in the Z line during muscle contraction, but the distance between two Z lines decreases. Okay, because of muscle contraction, will make this move, okay, to the middle. Are you clear? Z line will move towards the middle, okay, because of the muscle contraction, sarcomere becomes shorter. So when the sarcomere becomes shorter, right, this length becomes shorter. So basically, the distance between Z line also shorter. C is the M line or MDs. So M line act as an anchoring point for the thick myosin filaments. Okay, can you see that? The C. Okay. So there's no change in M lines. Okay, but the distance between two M line also decreases. Okay, because when you pull it, so it will become shorter also. Okay, try imagine this one becomes shorter already, so everything becomes shorter. So the distance between two M lines also will be decreased. Okay, I-band is a zone contain actin thin filament only, which is not superimposed by the thick myosin filaments. So the I-band becomes shorter during muscle contractions. A-band is contain the entire length of the single thick myosin filaments. During the muscle contraction, there is no change in the length of the A-band. Then F, structure F actually is a H-band. H-band is a part where only the thick myosin filaments are present with no superimposed by the actin filaments, and the entire length of the H-band become shorter. Okay. So with this, I've done for the first part of the lesson.